Good afternoon to everybody in Vienna. My name is Ines Caira. My lab is based at Queen Mary University in London, and together with Kevin Bird at the American Dental Association, we are coordinating the oral and craniofacial bionetwork. Oral and craniofacial tissues are incredibly diverse and include the tongue, teeth, gums, glands, and all these tissues are harmoniously integrated into the vital functions of communication, feeding, breathing, and early digestions. Our ultimate goal is to understand the common and unique cell types that support these niches in health and uncover which cell types and networks are affected in disease. And for this session today, we were asked to provide you an update of the roadmap of the Oral and Craniofacial Atlas. So this network was created in late 2020 with the need to map the oral cavity and understand how SARS-CoV-2 infection is affecting the salivary glands and the oral cavity. A great effort from the teams of Sarah Teichmann, Blake Warner, and Kevin Bird that was published last year in Nature Genetics. This was, let's say, the first official paper of our network, but several other studies, linked or not to the Human Cell Atlas, have contributed to the effort of mapping the oral cavity, including studies from the labs of Paul Sharp, Igor Adameiko, Niki Motopoulos, Anunia Opasawachai, and more recently from the CZI in the table of sapiens. Our network is still at its young stage when compared to lung and gut network, for example, with only 1.5 years so far. But we have made significant strides, including nearly 300,000 cells sequenced, representing the first cell types of 8 distinct niches from healthy adults, so buccal mucosa, gingiva, tongue, tooth, salivary glands, parotid glands, tonsil, and saliva. And while work from the oral and craniofacial bionetwork and other teams has begun to reveal the heterogeneity of diverse tissues and fluids of the oral and craniofacial complex, together with Sarah Teichmann with the team of Sarah, we are currently integrating data from the several published and unpublished datasets to build what will be the first integrated version of the Human Oral and Craniofacial Atlas, version 1, with common cell annotation for all datasets. So this will be just a first step in comparing the datasets, creating a consensus cell type annotation, and validating the existence of cellular subpopulations. And we anticipate that this version 1 of the Atlas will suffer from many of the analytical issues discussed before by other networks. With this, I would like to highlight the importance of having a dedicated computational biologist with a focus purely on data integration efforts, working directly with the bionetwork coordinators, as well as, of course, then the tissue specialists for the annotation and validation of the discoveries. Our network is committed to establishing the first draft of the Oral and Craniofacial Atlas within 2 years. And as our next step, we are planning to conduct studies to expand the tissues represented and how this is also affected in disease. And this will be the scope of the Atlas version 2 and beyond. While we did not benefit from the first wave of seed network funding to kick off our adult atlas, we were now able to secure funding from CZI, and we are contributing to the Pediatric Oral Tissue Atlas as part of the Human Cell Atlas Pediatric Networks by joining efforts with the Skin Pediatric Network led by Musanifa and the Nasal, Mouth and Airways Pediatric Network with James Haggard and Fabian Theis. And bringing a multiomics approach, spatial mapping of these populations is also undergoing and will provide further key insights into the tissue organization and, of course, the cellular crosstalk in health and then in disease. We have recently published the roadmap of our Oral and Craniofacial Atlas in the Journal of Dental Research, with the aim to gather interest from this community and to foster new collaborations. And of course, this atlas will then be expanded and compared to disease and to other systems and other models. So there is current effort in my lab to compare healthy human oral data with mouse oral cavity and skin data, as well as with disease affecting both oral mucosa, skin, and skin, such as lichen planus and epidermolysis bullosa. Our teams in the network are exploring other oral and craniofacial tissues also, such as the salivary glands and gingiva in the context of Sjogren disease and periodontitis. And of course, we'll be comparing with in vitro models and organoids and 3D cultures. So our next steps are focusing in large-scale integration of the currently existing datasets for adults, the generation and integration of multiomics datasets funded by the pediatric grants, and to deepen our biological insights into the different tissues and how these are affected in disease, and then on providing equitable data access and data sharing to maximize the impact and the utility of the generated data. So to achieve this, we will greatly benefit from a strong computational support, working across the teams to integrate the data, and ultimately to curate the data online to ensure the long-term use of the data sets beyond the lifespan of a PhD, as Fabian was mentioning yesterday. So thank you all for your attention, and Kevin and I will be happy to discuss this further and see you all very, very soon, I hope.
Yes. I should be able to now run the audio for Matthias on a different machine.
I don't know if I would be able to start tomorrow to start because really late for different reasons. And
Could please mute yourself if you are not.
So probably I will start then, not tomorrow, but the after. But anyways, you will get data in, as we discussed in time. So.
Thank you.
Please accept my sincere apologies. I'm not able to join you to provide a brief update of the gut network. Firstly, just a brief status report on progress with regards to our roadmap. The last few months, there's been over 30 members actively contributing to a manuscript, which we intend to submit to Nature Reviews, Gastroenterology and Hepatology. We've kindly agreed to commission this work, and they've given us a deadline, the 9th of September this year. So it's good to have a little bit of pressure on that one, but we're all excited about the manuscript. In terms of challenges for the roadmap, amongst many, defining the baseline requirements for what we would consider the first complete draft of the roadmap surely is a challenge, and probably not unique to the gut network, and certainly work that is ongoing. Practical challenges included coordinating contribution from a large number of gut network members who contributed to the writing and preparation of the roadmap. So finding suitable times that fit all time zones can be a challenge. And of course, sort of, making sure that we capture the expertise of a wide range of scientists and clinicians was also a challenge. In terms of the overall structure and sort of highlights of our roadmap, we of course tried to highlight unique aspects of the gut. And amongst other things, we highlight our aim to really combine data, not only single cell sequencing, but also spatial data, imaging data, extensive metadata, and in that respect also highlight the importance of documenting and describing clinical data for human tissue samples that are being subjected to single cell profiling studies. We also aim to develop and apply a common coordinate framework, again, trying to integrate several data sets and multiple methods. Another important aspect of our roadmap that we want to focus on is to show ways how we can integrate the gut roadmap in health with related diseases, for example, inflammatory bowel disease and cancer, and also integrate the gut bionetwork with other networks such as organoids. In that respect, we recently held a joint workshop between our networks, which I thought was really quite exciting and productive. In terms of data sets that we are planning to include, we've identified quite a large number of relevant and high quality publicly available data sets, just over 50 or so, and we're still working on selecting the most relevant that we ultimately then decide to include in the roadmap. And our roadmap manuscript will include a detailed table and description of the top data sets and where to find them and how to apply the data. What are our bionetwork's computational needs? Generally speaking, in the first instance, and again, it's probably similar to other networks, we benefit from support with combining different data sets in terms of atlas. A key aspect is also annotation of the data. That's both regarding annotation of individual cell types, but also integration of metadata, and perhaps help with performing adequate, appropriate, and standard QC checks of these data sets that are available would be really helpful. At a later stage, we would also benefit from help with integrating other data sets. As I mentioned, we aim to integrate spatial data, but also multi-omic data sets such as epigenetic profiles, single-cell ATAC-seq data, and if available, also relevant data sets such as adjacent mucosal microbial profiles or imaging data. And this is about it. I'm just within 5 minutes. Thank you all for your attention and hope to see you all soon.
Yeah, are you next, Jack? Okay.
Everybody. First of all, let me just check are the scribes here in the room, just to identify yourself. Yeah. Hi. All right. So okay. So just as a show of hands, so I know who is who. So who here belongs to the or works as a part of the gut network? Okay. And the pancreas. Good. And the oral and craniofacial. Okay. All right. Okay. Oh yeah, we're online. Of course we do. Yes. All right. Hello, folks that are online. Let me just, if I do it like that, can you see them more? No, I don't think it really has any effect. Okay, never mind. Okay, so Hang on. Okay, so I put together here a list of topics for us to discuss, and you can just kind of go through them, and I want to engage you, to encourage you to have a conversation and just speak your mind and so on. And for me, I can tell you, giving my own perspective, it's really interesting to hear what people here have to say, because it just gives me some insight to where these large projects are going and where are the needs, and that it already came out multiple times in the introductory talks that people gave here, right? So, but let's dig more into that, right? So I have a lot of questions about it, right? So we don't have to go through all of them, but the ones that we feel are engaging and are necessary. Oh, and I forgot to mention that we have Shayam here, who will be in charge of leading the diversity discussion, and I put it as a part of our discussions here, and we can just put it out there or in any time that you see fit. Okay.
Thank you.
Okay. So yeah, so the topics that you see here. So first of all, we can talk about some of the annotation efforts and the challenges there. Then about the sufficiency of the atlas. So what people think here, based on what you know about your network, about what people are collecting, what is there, what is missing, diversity. Then thinking about metadata again. This thing came out already in the talks that people gave until now. Using the atlas, so it would be interesting to actually do with the atlas once it's there. How do you collaborate? What are the means? How do you share data? How do you talk to each other within the network? And issues of computation that will come up, I'm sure, throughout the subject, but anything that is left after that. And of course, happy to listen to additional questions and so on that people have along the way. By the way, people that are online, can you hear me okay?
Yes, thank you.
Yeah, oh, and we also have Rick here, who is right, he's in charge of the computation. Rick in the GATT Atlas, right? Did I get it right?
I think in the integration efforts in general. Yeah.
Yeah, okay. So yeah, so let's start with the annotation part, right? So I'm curious to hear from people here. What are the challenges you came across doing annotation? So let's start with that very general question, right? And then we can dig in. Please.
I think one remark that I'll make is that even in high impact journals, we have some issues of annotations.
Issues of bad annotation.
Yes, so very mistaken.
Absolutely. Yes, that's definitely. So what do you think can be done to correct this?
Perhaps an erratum. Someone would have to bring the.
You mean adding some
sort of correction.
Correct. Okay. So yeah, I guess so. This is something that came up earlier in the sessions today. It was in the morning. The morning seems like a long time ago, but yeah, we did see the cell annotation platform, right? We also have this machinery in Tabular Sapiens. I think this is something that's a very important point, right? For some reason I didn't think it's going to be that prevalent, but I'm hearing more and more that it is, that people see things that are anywhere between like a gross annotation error or something that is maybe very specific, and maybe this cell, maybe the boundary and so on. So I see that there is a lot of discussion. So having platforms like that, right, that makes a lot of sense to me or something. But maybe going back to the network, right? So as somebody who generates data, right, you have this you put it through, you have this matrix of set by genes and so on. How do you go about annotating yourselves?
I think it's a very kind of simple overview to what we've tried to do. So we focused a lot on building hierarchical structure. So basically doing iterative tiered clustering. And we actually ended up developing an automated algorithm that helped us to cluster 2 points that were deemed computationally stable. And actually, if we didn't do this, we ended up with a lot of cases that may lead to misannotation, because either you were under or over clustered. And so I think that one thing that we maybe haven't talked enough about actually is how clustered is your data, and how do you know that that is the appropriate clustering, right? The example was given that if you give it to 10 people, you'll get 10 different answers. And then I think this is a first very important step, because oftentimes when we're doing the annotation, it's going to be relative to a certain outgroup, right? So you're annotating, for example, an N cluster. Is it relative to gene expression of all cells, or to its parent cells, or to its siblings, and so forth, right? And so I think that a cell can have essentially that many identities, and so actually building out these hierarchies will probably find consensus up to a level, but then there needs to be also room for kind of free form interpretation in some way, right? So this has been our approach in general. So fitting cell ontologies up to a certain level, but then letting us also try to say, hey, maybe we haven't seen this before, right?
I see. So trying to look at the data at different levels of resolution, both in terms of the ontology and the data itself, and stopping where you can't make a decision anymore. Yeah, that sounds cool. So any other approaches here people want to mention that they're doing with their data?
The other thing I'll add is actually, since we're now working at a fairly sizable number of patients, is we don't filter out based on actually clusters that end up being patient specific, but we do flag them in our annotations, right? I think that, even if you're working with data sets of 30, 40 patients, it doesn't scratch the surface of, maybe total gut diversity, but rather we basically apply a diversity metric of how many patients are represented within that N cluster, right? So that we understand, is this something that's conserved or not? Because even things that are very low in number can sometimes be quite conserved, and things that are very high in number can sometimes be very patient specific, depending on the cell type.
Yeah. So the people see, go ahead. I guess that connects to the larger efforts here, and I think probably the gap with the pancreas as well. I saw in the pancreas that there are like 14 different data sets that are being put together and so on. So what are people's experience in seeing those differences, right, between different annotations? How do you mitigate that? What are your, do you perceive it as an error, or is there some procedure to see whether this is really diversity between humans? As for this, any thoughts about that? Okay, so
Yeah, you talked about genuine patient-to-patient variability, where you can't just assume that they have to land on top of each other. And generally, in colon cancer, we saw that too, that if you force them to land on top of each other, all you're seeing is cell cycle variation, and you lost all the biological differences. An interesting thing was how they were different. So we actually decided not to use data integration across patients. But then I guess for the healthy samples, perhaps it's more justifiable to post them on top of each other. I don't know what you think.
How is the data brought together if you were not? And were you comparing from the same study, or
Different studies.
so how, like at what level did you?
For each study, we zero centered each gene.
Okay.
And equalize the data. That's the. We still saw the patient specificity, but some other weirdness is clear set. But of course, there were batches.
So that means that, but between let's say phenotypic groups that you can stratify very clearly, so there you prefer to keep those separate. That's what you're saying, because you felt like integration was too much. Yeah.
It was clear there were biological differences between the patients. So
yes.
You don't want to lose that.
Yes. Okay. Okay. So here is another question that might be interesting to discuss is how good is the current cell ontology, right? When you have your vocabulary, when you want to look at the pancreas, for example, or at the gut, to what extent do you feel like you have access to cell ontology that is satisfactory at the moment for your interpretation? I see smiles, that means that not so much.
Yeah, I don't know. I mean, I think it goes along the same lines that up to a certain point, I think the ontology is very good. And I think we can find an easy consensus. But then it gets more blurry, right? Because also states are not binary anymore. And but that's also where it becomes interesting. So
yes.
Yeah, and there really I think we have to find a good way to also communicate if something is wrong, or if there is a good reason of the authors stating that this state might be relevant or something.
Yeah.
This is kind of major challenges.
One thing that we've also found helpful too in our kind of clustering and annotation efforts is actually the way that we cluster is then not how we annotate. So what I mean is that we basically cluster down, and then once we have those end clusters, they naturally group into kind of T cells, B cells, etc. But then we build a tree for each of those, and that tree gives us essentially the linkages, right? And up to a certain level, each branch of the tree really fits nicely within the cell ontology, so we can append that. And then we get into some, more maybe unclear states, etc. We can then give names that are basically based on genes, right? But you have the ability of ontology with state, right? And you can kind of blend both. So we've tried to do this, but it's important to make the distinction between the clustering steps and the actual tree building steps. And I think actually the brain's done this fairly effectively in terms of what I've seen from Alan as well.
Yeah.
Yeah, I think that's a valid strategy because I was in the annotation group. It felt like there is an interest of having a really comprehensive list of terms. So we at some point to have to deploy markers of a given cell type to branch it into subtypes rather than keep creating terms.
We have to move toward consensus about what those terms mean. Like, for example, the transit amplifying cell, right? You have one data set where it's 70% transit amplifying cells from a biopsy. And then somebody else does a similar biopsy and their transit amplifying cells are only 30% because the definition of what's a transit amplifying cell versus a stem cell versus a mature cell are different. Right. So yes, you can have the terms, but you also have to agree on what gene signatures fall within those terms.
Yes, and you think that there is
but this was with single cell RNA sequencing data, and before that with lineage tracing was also the semantics between what's a stem cell, what's an active stem cell, what's a quiescent stem cell. That's always been a matter of discussion. So hence the importance of these jamborees when we get together to have a consensus, not only annotation, but also a nomenclature for each of the cell types and states.
Do you feel like that? Oh, please go ahead.
I was just going to say, is this not also a question that could partly be answered by integrating data sets? And for example, like with the lung that Malta and Fabian have presented, that they have these kind of correctly annotated, incorrectly annotated, under annotated. If you pull all of the data together, and then you can see, you annotate the reference data, and then you take the original annotations attached to each cell, and then see, was the original annotation the same as the consensus annotation from the integrated atlas? And that can be something that everybody in the network can contribute to in terms of coming towards this consensus. And then it gives you an idea of, okay, actually, transit amplifying cells are misannotated frequently, and then people can be aware of that when there's new data coming out.
I was going to say that I think in those efforts, you gain some power in discovery for rare cells.
Yes.
But you actually lose in terms of things that are gradients. And so what they noted is at the same time that they can find ionocytes and neuroendocrine cells, you actually lose resolution to cytotoxic T cells. And now you can no longer differentiate them from NK cells, right?
Yeah.
So actually, at what level you integrate becomes very important, because in some cases you can over integrate at a high level and you lose your ability to find things that you know are there.
Yeah, but I think that's also a really great point. And then also taking, so the reason I'm talking about the lung is a common theme in people taking examples from, but I also come from the lung, I'm both kind of across networks. In terms of the lung, we have an integrated atlas where they gain things for certain cell types, such as red cell types, as you said, and then lose things for, you said cytotoxic T cells. They also don't annotate Tregs in that,
Yes,
which is,
and
it is in general, like for T cells, it is a challenge to identify different clusters because we don't have that, classic flow cytometry, CD4, CD8, unless we have cytate, etc. Anyway, the point that I wanted to make is I think you need both integrated large scale, because then you have the power to start looking for these really kind of complex metadata trends, BMI, age, what have you. Then you also need more focused studies that use a really comprehensive sampling approach, like a study that I've been heavily a part of, where we took 5 locations from the lung. We did single cell, single nuclear, spatial. We're going to talk about it tomorrow, but really, like, then you can control for these batch effects, and that's the reason that you use these kind of murkier cell subsets. Then you can have dedicated, really well curated studies on few donors, annotate really well, and then have these larger integrated atlases. So then when new data is generated, you refer to both to annotate your cells and figure out what is there, and then see how it fits into the bigger picture and your own biological question.
So again, the separation that you're doing between these annotation, let's say cohorts, is it based on the individual sample, or like Shyam said, it's also based on the context of the sample, right? Or are these all comparable sample in terms of can these be seen as replicates, or where are the differences between these samples? Where do they come from, and why is it important to keep them separate?
As in, like, having like integrated and then
Yes,
dedicated.
yes.
Yeah, so I think it's because if you have a dedicated, really well thought out sample study, you can control for batch effects much easier. I mean, you still have batch effects, but you can control for them a lot better. The other really key from this study, like a focus study that I'm talking about, is that if you combine from, for example, the same donors or the same kind of lab processing, controlling these batch effects, things like single cell, single nuclei, different digestion enzymes, all these things will affect your cell composition, a lot of different variables, and that's why you can then have a dedicated, well-planned, thought-out study for granularity, and then the bigger integrated, and so this study that I'm talking about is what Malta and Fabian have shown, where they have the core HCA, and then they map this additional study. This is the study I'm talking about, Madison and Oliver et al. And there they demonstrated that there were cell types in that study that weren't present in any of the core atlas studies. And this, some of these things were like erythrocytes were one of them, chondrocytes from the cartilage. That came from single nuclear data because we didn't have the bias from digestion enzymes.
Oh, I see. So they were actually not present there. It's not because,
it's not because people under annotated or anything, they just weren't there at all.
I see.
So then you also have under annotation and things, but there was quite a few cell types that just weren't present at all.
Okay, so that's almost sounds like a call to arms for the computational people to make these things work better, because but I do agree that there is a limit of how much you can integrate, and at some point things start to break because there are these differences either in the protocol or just in the biology of the donor. So that makes a lot of sense. Yeah.
That was almost answering the fourth question there.
I do. I want to move on to the next slide, but I want to ask one more thing. How do you organize the tissue? So I can tell you, I can give you a small, short and sad story. When we worked on the Tavia Sapiens project, so we actually had a lot of debate whether to look at the tissues, to stratify by tissue or to stratify by compartment. And we've learned that actually stratifying by tissue ended up being much more effective.
What do you mean by compartment?
By let's say immune stromal. Right. And so I'm just wondering, what is your experience? What are your thoughts about how do you organize your tissues in that sense? Right? How do you take the matrix apart for one tissue?
I think it works a lot easier by doing the way you just said, including like, especially when you're doing downstream analysis, regulatory elements, you don't want to have immune cells and T cells.
Well, actually, we actually end up working by tissue, right? So that means that you have them together. But you're doing the.
Yeah, I prefer to separate
by compartments,
by cell types.
I see. And any thoughts about pros and cons? Because this is one of those decisions that sound a bit technical and so on, but it can have a lot of influence. So that's why I'm specifically curious about that.
It's also very, like, the way you process your sample will bias what type of cells you get out the other end.
Yes.
Right, so you can have a prep that'll give you more epithelial cells than immune and vice versa. So you kind of have to know the question that you want to ask even before you digest your tissue.
Yeah, I think doing cross-study frequency-based analyses is very dangerous.
Yes.
Oh yeah. Oh, that's
Yeah, you can't.
yeah.
I've seen this.
Yeah, you know, and you know, it's something like within the Helmsley network, we talked a lot about this. Like, do we standardize from the beginning and make everybody do it the same, or do we accept the fact that everybody's going to do it differently and deal with the problem later? So we chose that second, and now we're up to the deal with that later.
Yeah.
But you know, when we started this 3 years ago, there was no consensus on what the best way to do this is. I don't know if there is now, but
that
there certainly
speaks,
wasn't that.
in terms of this question, that speaks of going by compartment in a sense, rather than by tissue. If you do want to do some broad integration analysis.
Yeah. Actually, the integration we found is working better by compartment, by cell type, as you mentioned.
Yeah. Because of compositional differences,
Yeah.
they have much more effect. Yeah. I can see your point. Yes.
I have like a question, but also I want to point out that someone is trying to use like live transcription to enable that. And then possibly it's really possible that we still could hear discussion how. Okay.
So if it's by compartment, it allows you also to look into the similarities between the tissue. Wouldn't that be easier than if you look between different tissues, between different tissues?
Yeah, that is correct. That's definitely it makes sense. Yes.
So now my question, this might be like a data usage kind of question because we're in the portal for business vectors, cell by gene, the HCA, DCP, DCP. So is there interest, if you're looking for data to cross compare, is there interest to be able to like search by compartment, or do you want it at the tissue level? Because we're like, some of our tools are able to support like immune system or like research domains. Would that make it easier to find data?
So now I'm going to repeat the question for the now. I learned my lesson. Yes. So a question here from Cell by Gene.
Cell by gene, HCA, DCP.
Or from the HCA, DCP and Cell by Gene about how to stratify cells when they are being made available, let's say through a Cell by Gene session, either by compartment or by tissue. That's true.
Both are interesting to look across, you know, the GI tract, for example, and compare different locations, and then also to compare different compartments. Yeah. What's like, if you had to pick one or the other, I mean, ideally both would be made possible, but if you had to pick between one or the other, which one? Maybe at the tissue level. Yeah.
Both.
Yeah. Okay.
I just wanted to add to the last point of the annotation also. It would be quite interesting to do this. I think this came also out from the food talks from yesterday, to actually group them. I mean, immune cells, stroma cells, and epithelial cells. If you go into the spatial, universe, I would say that they are grouped together, and this might be also a good way to compare different tissues, where you have these composition of different compartments actually, and this might be also patterns which arise from, for instance, in disease, inflammation, and so on.
In spatial data set, I agree with you, because there we have much less concern about the composition. I mean, I'm sure that we really have some biases, but not as much, right? Thank you for this point. Yeah. Okay, so sufficiency. So I know that the networks that we are talking about, and I hope that I got it right, but it's multiple projects. In some instances, it could be multiple groups So do you feel like where things stand right now, do you feel like you have sufficient amount of information to build something that you can call a draft atlas? What are people's thoughts about that?
Well, I'll just kind of expand on what I said earlier, which is that, like, especially for pancreas, we're having trouble tracing back individual samples to get all of the necessary metadata. So there will be a data set in the portal, but it's got 14 donors in it, and we don't have the demographics or the metadata for all those donors. We don't know how many cells go to those donors. We don't know what split of those donors are healthy versus diseased. And so it makes it challenging to assess how our coverage is, how our diversity is, et cetera. So I think that's one of the challenges of the like current model of data upload and data repositories, is that it's broken down at a study level and not a sample level.
I see. So you would ideally like to have more of this metadata, being able to see it in those repositories to make the appropriate, yeah.
Hand up.
Thank you, Liz. Yes. Yeah, Karen, please go ahead.
So you mentioned that you have different data sets also containing healthy and diseased. Are you planning to also add disease data sets as part of your reference? And if yes or not, what was the reason for the decision?
Yeah, I mean, for us, we're trying to separate out just the non-diseased for the beginning. I think ultimately where everybody wants to go is to be able to compare and analyze the disease, their various disease states. But I think we have to start somewhere, and so hopefully it'll make our lives a little easier to just start with non-diseased. Maybe.
Yes.
In the case of the oral atlas, we are following the same approach, even if some of the samples were harvested in parallel, so controls, and then, for example, periodontitis or buccal mucosa. For the first atlas, we're going to focus in healthy tissue, and then using this as a reference to map the disease tissue and the pediatric as well.
I was just going to ask about how you define healthy in this context.
Yeah.
Right? Because if it's matched normal from the same, a different part of the same organ that's diseased, for example, and there's an annotation for it, that's the difference between true normal versus fair normal.
Just to make sure that you heard the question, Karina?
If you could repeat, it would be great.
Yeah. No, so I think the question was, how do you even define disease in this case, right? It can obviously be a very subtle spectrum and different conditions that you can consider as flagging something that you separate in samples. So what are your thoughts about that?
Most of the times we always get some histological sections to compare, but then you will be able to rule out, of course, any kind of inflammation that will be, well, in gingiva, for example, it's common to have already some inflammation even in a healthy individual. What we will consider macroscopically a healthy individual. And then, for example, some of the samples that we were gathering or from people saying, well, we can have adjacent normal that is adjacent from a tumor, to what I say, of course not, because there's always this field cancerization in all the tissue around the tumor that histologically can look normal, but we know it's not normal and can already be carrying some of the mutations that will be the founders for the carcinoma. So we have to be very careful about how to sample these. And for us in oral cavity, it's been quite a big challenge to be able to gather healthy tissue from people that are not undergoing any kind of treatment. While in skin, we were easily able to get surplus from mammoplasty or abdominoplasties. In the oral cavity, it's very difficult to get a volunteer to give a bit of a tissue from their tongue, for example, while buccal and gingiva, that's much easier. So we are facing that challenge, and in UK, we are now working to try and get post-mortem tissue, but I know that in other countries that's easier than here. So we are likely lagging a bit behind on that sense. but in the tabula sapiens from the CZI, for instance, when they got several tissues post-mortem, then tongue was one of them. So we are working on that to be able to increase the samples of truly healthy tissues, making sure that there's no underlying disease or inflammation in the tissue.
Yes, please.
Can I add on to that? Sorry, I'm just going to move my mouse so just really quickly, I wanted to go back to the point about metadata. I think that's like across the board an issue. And as I said before, I think one of the interesting things of integrating lots of data sets is to look for, like, have the power to look for these differences. If you have 1 million cells, but only 100,000 or 200,000 have this metadata, then you can't do the analysis. Then the second thing about what is considered healthy, there was a discussion about this in one of the breakout sessions about what makes good Atlas, like the breakout 1.1. And basically, I think we're limited, as was brought up, for where we can get the sample types from. But when we integrate, hopefully the idea is that, you know, we can get, for example, maybe samples from deceased organ donors who don't have any underlying gut or pancreatic disease or what have you. And that is the most normal that we could possibly, like, healthy that we can possibly get. Then, you know, you may have some neighboring non-inflamed or implicated areas in some disease reception or what have you. And that's between the spectrum of disease and healthy. Then we have true disease data. And so when we integrate all of that together, hopefully we can see that progression, and that's interesting in itself. So, you know, as long as we have that information, it's actually really powerful to have healthy and kind of healthy and disease, right?
I might add to that the metadata there for the patient health to disease spectrum needs to be very linked to the metadata for how it was collected, because typically an autopsy-based study or a screening colonoscopy or a disease colonoscopy may come through 3 very different routes, right? And so those have 3 very different types of sample processing times, etc. And I thought a lot about this for the gut, for example, and you think about someone who is in the hospital and then they have tissue collected afterwards from an autopsy. They probably have not been eating normally, actually, for quite a prolonged period of time, right? So all of the standard types of nutrition that we experience is simply not there, right? So there's all sorts of caveats with this. And I think the only types of real healthy tissue in the gut right now are probably 60-plus-year-old screening colonoscopies.
Exactly.
And then there's a big gap, and then you have fetal gut, actually. Fetal gut might be a kind of, and then there's basically very little, right? In terms
In
of
our Belgian-Swedish consortium, we actually have real health volunteers.
Yeah.
So these are subjects that have absolutely no symptoms according to clinical criteria, laboratory analysis as well. But of course, we lose a lot of potential candidates because of this level of screening.
Yeah.
But also screening colonoscopy is also with a PrEP, right? Which is also
Yes,
something-
exactly. There's the washout and yes.
So
I would also just bring up the point in general, I'm sure we're all aware of this, but there is no perfectly healthy human being. We are all along the spectrum of health and disease, right? So the point is that what we can collect isn't representative of that full spectrum. But, you know, we can try to do our best, and things like having people volunteer to have biopsies, you know, can help.
Yeah. So I guess in the what is missing, if I can summarize it, so we talked about the level of information that we have on the samples that we do have, but there is also a certain amount of resolution that, or information that we can have about that, right? Just because of this, it can be even microscopic differences that are there, or things that are more progressed, but they've not been observed necessarily, things that are much more solid like diet and so on, and all of these have an effect on whatever it is that we're seeing in the data. Yes, please.
So if you wanted to like understand more, when you're using someone else's data, if you wanted to get a better understanding, do you like reach out to the lab? Would you expect to find that in the paper, like I'm describing our definition of normal mean?
Yeah, I usually start with the paper manuscript if there is one, and oftentimes that gives you enough information to at least kind of group it.
Yeah.
Usually there's a supplementary table.
Sometimes
Sometimes
you're
there
running
isn't.
to
Yeah,
privacy
the
issues.
only option is to reach out to others, and that is a lot of work.
Yeah,
If, you know,
and not always successful.
Oh, interesting.
Yeah, exactly.
I just wanted to add also to the Pancreas bio network. I mean, from the European HCA project. I mean, we did this by organ donors, and we have now from different cohorts, fetal, isolate, type 2 diabetes, and I mean, all is also done with different methods. So from different angles. So we have single nuc, ATAC-seq, and also proteomics work ongoing. So with 500,000 of single nuc RNA-seq, I think the Pancreas is sufficiently, or it's ready for draft 1.0. And if I would now just, you just said this also, there's probably no real healthy pancreas in my age, which is most of the donors' age. And you can just add other age cohorts, or you just do more technical advances, which could be, proteomics or in combination with other genomics and transcriptomics methods. So I think we reached already, and maybe also Will and also Timo can comment on this, but I think we reached the point of sufficient data in Pancreas.
Okay. Interesting. Well, that's a good point to make. Okay, so folks, I just because we need to wrap up by which by 5:45. So
Or
5:50. All right. So okay. So I guess let's move on, and then we'll see if we go back to that. I do want to have some time to spend on these other subjects. So let's help with diversity.
We should also spend maybe 10 minutes at the end to sort of synthesize.
Oh, okay. I see. Okay. So in that case, we've heard some, maybe a bit all over there are some opinions here. Is your opinion that what we have right now, right? Thinking about the pancreas, thinking about the brain, we feel like, going back to that question, do we have enough to say this is an integrated atlas that gives us a helpful, a useful representation of that issue? Yes.
Sorry, I'm sorry if I'm saying a lot.
Yes.
I actually would really like to, I think this is like a really nice combination of networks, because the theme is digestion, right? Like all of these organs and tissues are involved in digestion, and among other things. But what I would like to maybe almost advertise is, as a representative from the gut team in the Typing Lab, we are doing some of these integrations for the gut from fetal and healthy, and we haven't just focused on like stomach and intestines, for example. We also have esophagus. We started to include oral, dental samples, pancreas, gallbladder, all of these things. So I guess my question is, do people think that it's useful to have every kind of organ tissue involved in digestion in one atlas? And then I guess each individual kind of sub-network is also working on a dedicated atlas for pancreas. Is that useful? Should we work together more? Because I feel like it potentially is disjointed.
Oh yeah, question to the audience. That's actually one of the subjects we're encouraged to ask you folks about this host-tissue integration. That makes a very makes a lot of sense.
Is this like from the tabular statement, like from the
donors or you have
No, so it's like, the integrated lung atlas where we're trying to find, we kind of looked for studies, and I think, we have kind of advertised this in some of the gut bio networks, and Matthias in his presentation mentioned this joint, gut and organoid network. Raquel Oposo from our lab talked about these efforts. But yeah, I think I'm realizing, because I've come from the lung and now also involved in the gut, I'm realizing that it seems like a lot of people are doing a lot of things, and it would be good to come together a bit more. And this is a great opportunity to mention this and to put this forward, that, we want to make it a community effort, not just, one or two labs are working together on a project.
So which organs do you think would fit in that?
Well, we have what I like to call the oral cavity to rectal package, plus pancreas, liver, bile ducts. If people think of other things that are useful to include, esophagus as well. I mean, I guess that's in the oral cavity to rectum package.
That's in the video.
We're trying to include everything as a comprehensive reference, and then it probably also makes sense to have more specific, because then you may see things that are lost by batch effects and what have you in this mega atlas. But it's challenging with metadata, with cell annotations, all these kinds of things. So it's not just one group or one person should be doing this. It really needs to have input from all sides.
Yeah.
I think we also the gut is incredibly big, right? So when we at Helmsley decided to go after this, we decided from the beginning just to do the terminal ileum and the ascending colon. So we're the big bear in the room, and we're skewing all the data toward that. But by the same token, we will get really good representation of those two parts of the gut, and then hopefully that'll be useful for the rest of the colon, the rest of the small intestine, and eventually everywhere from mouth to anus. Just a little.
Well, actually, so that was actually the original term that we coined, but I thought in a more professional setting it's nicer to say oral cavity to rectum, but let's go with mouth to anus.
That's how we describe Crohn's disease. It can affect you anywhere from mouth to anus. It flows freely from the tongue.
Yeah, but Nir, have you done this, for instance, intestine, just to integrate this data? I mean, from the tabular. I mean, is
integrate with? You mean with the with mouth to anus in the tabula sapiens?
Yeah.
No, we have not. We I like this. I'm just gonna make sure not to use it in the but no, we haven't, and that's really good. I mean, it makes so much sense, right? Because there are no barriers, right, in that package. So that's definitely makes a lot of sense. Yeah.
And I like the idea. I mean, to what you call it, rectum package? No, I mean it is. But it is I think a good idea. I mean you try to merge or to join, assemble, and let us say in a body function or something, and just look into. I mean, obviously, I would not do any kind of manual annotation in this, but I mean, with all these, with this popular vote method or with any automated annotation, would be interesting to see what comes out of it. Yeah. I mean, this could be.
Annotation obviously takes a really long time, but we also have tools now like Azimuth and cell typers, which really help. You still need to put a lot of work in to make sense for cell types like immune cells, etc. And it will always depend on certain hypotheses that you might have in mind, but it's still. I think annotation doesn't take 2 years anymore. You can use these tools, and we have enough data to feed into those tools that it becomes easier.
Right.
So I think it's very useful to think about these gradients, right? Especially if we integrate with the people think about, even the Latin colon, which are the different development origins, remain poorly defined. And this is the prior to what was happening in the mouse and the central community, where people are now starting to make microdissect the entire thing of the colon and the small intestine and using the sequencing in every single part of that. But that, of course, to do this in human setting requires detailed annotation, which is going to be a problem. Right? And even, just from the clinical standpoint, just even getting annotations of where from the colon the clinical gastroenterologists and surgeons take the sample is really challenging. So how are you doing that?
We don't know yet.
Yeah. I mean, the problem is that there's basically two ways to do it, right? If you're doing a biopsy, you can say how far has the colonoscope advanced, right? But everybody's colon is different. So yes, that gives you information, but it doesn't necessarily allow you to compare it. You can also use landmarks, right? It's between, right? This flexure and that flexure, halfway in between. But again, it's really vague.
And not all the data sets are annotated.
Right, but even the ones that really try to be annotated, you don't get the detail, you don't get the really fine detail, because if you ever looked at MRIs of colons, they are radically different person to person. Like you see the pretty pictures and you think everybody's the same.
Then I think it's also up to the community to then keep this in mind going forward. Can we go away, think about what may be the best way to try and come up with, like, for example, in the lung, they have this common coordinate framework. I think someone's going to talk about that later. Can we try to equate that or adjust that to make sense for the mouth to anus? Or yeah,
We started that, right? So the Albert Berger in Edinburgh has been starting with Mark Ahrens doing a common coordinate framework. And again, we ran into this exact same problem, right? We have we go from rectum all the way up to terminal ileum, but how do you then map your individual samples onto that? It's essentially guesswork. Eventually, what we're going to have to do is come up with a molecular signature that actually differs enough that we can place things molecularly in their correct context. But the only way to do that is basically to take the gut and sample it like every millimeter or every centimeter, which is a huge undertaking because 30 feet. Imagine 3.
And the compromise, there are some studies in that direction, but the compromise is then they don't have enough cells.
Right.
So they use the rare cells.
So this is why we start with version 1 and go from there.
Yes, so I guess one is missing, right? So I guess you have bits and pieces of this entire system on entire package itself. So of course there are all these still unknowns in the middle, but it's definitely encouraging to start thinking about it. It makes a lot of sense also in terms of describing the data, right? Defining commonalities. And yeah, so that makes.
And it also just one really last
Yes.
point, sorry for taking up space, but it may be okay to sample, just a bit here and there, but until we do some kind of hypersampling to definitively answer that question,
We'll never know.
Yeah, exactly.
Yeah, absolutely. And that's a challenge to do.
We actually do have within certain of our projects the idea that you take a surgical sample and then you sample along it just to try to get a gradient.
Yeah, oh, yeah.
Okay. You can't ask too many people to do that because it's just too expensive.
Right. So so let's move on here to our next topic, which is the diversity. So the first question is, knowing your data, knowing the data from your network, do you have a sense that what is your sense about the amount of diversity in terms of age, genetic background, ethnicities?
The answer to the second question is no.
We talked about
it.
That's a big one. That's really important, right? Very important.
I think in terms of like geographical and ethnical diversity, it's the same as all of the data sets. Generally, it's kind of like Western European American data sets reflective of the population there, predominantly white ethnicities. Ages, I think in general, it's a challenge to get later development, for example, like third trimester. I don't know if that's something that anybody is looking at for that. I know for other organs, people are working on that. And also, I think pediatrics can be a challenge as well. I'm also not sure for, you know, the organs that we're discussing how much data there is.
I mean, we have fairly extensive coverage of 6 to 18 years old.
Okay.
In our cohort, but it is completely lacking the genetic and geographical diversity because it's really it's a subset of a subset actually of families, right? Parents of the patients who are agreeing to have this procedure done for research. And so it is very much skewed white. And we are looking at people who live in rather large cities with a large academic medical center, right, that can do this.
Yeah.
And so, you know, I think one of the,
So
I
I guess here the question is proactive, but what big steps you think can be made within the framework of the networks that we have right now? What are your thoughts about that? And I know that there was some big talk about this today.
Yes, I think there are other people who are more knowledgeable about this kind of thing than I am. But a key thing is to have researchers in the countries that have predominantly ethnicities that are missing.
Yes.
And establishing better networks in those countries is a key thing. Because there's also, and this is something that was talked in the breakout session 1.1, but you can have ethnicities represented in, you know, a diverse city like London, for example, but you also have the interplay with the environment that may be associated with that ethnicity in some cases, and then you have like environment genetics. How does that interplay together? And that's also an important, I guess, geographic is there, and that's also important because I think you can try to raise and target certain ethnicities within big cities where there are these research centers. That's one thing, but then the next thing, and possibly a very important thing, is to also increase ethnicities geographically, or even independent of ethnicity, just geographical locations to represent environmental factors.
Yes, please.
Okay, so was definitely a consensus when you asked if metadata is sufficient for diversity analysis. So I'm very interested in understanding where like the metadata gap is. From my understanding, like a lot of like genetic and a lot of donor-oriented information is usually managed access. So are you going through the trouble of like requesting access, getting approved, downloading the metadata, and then finding that it's not as annotated as you'd like, or enriched or curated, or is it what you're searching that's limited? The resource or portal has to hide some of that information and only make it accessible for people that get access to it. That's a very long question.
So that question was about access to metadata, right?
Yeah.
Not about the diversity, right?
Yeah. So
Okay.
like donor metadata for, I guess, diversity or genetic.
I see. So you're saying that is a part of it, right?
It
So
might be like protected.
yeah.
The other issue is a lot of the ethnicity data is self-reported.
Yeah.
And you don't know what that means. We don't have any consensus about what that means scientifically.
Yeah.
And I add to this, it may be possible that we need very minimal metadata, right? So age and sex, I presume everyone has that, right? So let's check those boxes. Geography, at least we know the collection site. The patient may have traveled far and back, something is wrong with the response, but still we have a good idea for the collection site. So that leaves ancestry. Ancestry can be inferred from the data. So I completely take your point that self-reported ethnicity and so on is very fuzzy. But we don't, it's not essential. It's inferred from the data. So is it fair?
Can you explain that?
Like elaborate on, especially like if there's a minimal data size that that you need in order to do that, or type of data.
And the genetic information.
So just looking at SNPs in the single cell RBC reads, you can infer the answer.
So you collapse all the reads together, right? Because you're not really interested in gene expression.
So this also raises the point of data access, which again was something in the breakout 1.1.
Oh.
There was actually a point that someone made, which I thought was great, that there are ways that you can take out the SNPs and kind of de-identify the raw data, which is really important for integration methods because it's the best way to make sure that everything is processed in the same way. So then they're competing issues because you can remove, I mean, you can get the ethnicity information from the SNPs potentially, but then you're going to lose out on some of the data sets that are out there because ethically and the regulations of the country or the institution will not allow that information to be passed on for any reason.
Absolutely. So that depends on access to the raw data. So the fallback position is we send the software to the site where people are generating the data set. You run the software, infer the uncertainty for people.
And they're willing to share this information.
Just share the inferred uncertainty.
They're willing to
share the
share.
raw data.
I see. Just instead of the donor reported.
Yeah, exactly.
One additional point which came up from the integrated lungs that was some of the feedback on social media is, and this is across the board, I think a potential problem and misunderstanding of how we as scientists should be representing data on ethnicities. For example, a lot of the data that you get online for ethnicity metadata is like white, black, Asian, other. That's really insensitive, right? I mean, yeah, black is not an ethnicity, it's a race. I mean, maybe I shouldn't be having so much of an opinion as a white woman, but yeah, anyway, someone else can.
Yeah, this comes up all the time in these discussions. And for various reasons, various cohorts do collect that information and give you some course knowledge of how diverse is the data set, but it's not something you're using in any data analysis downstream, any molecular analysis downstream, because the genetic data inferred ancestry is more precise. So if you want to do any genetic analysis, you do the data analysis. You won't take the self-reported data. Yeah.
If that information is available.
So that's our strategy, at least in the genetic diversity network.
Yeah.
There are multiple teams in the Human Cell Atlas developing pipelines. Everybody knows it's in theory possible. So developing software pipelines to infer ancestry from the sequencing.
And you'll share those software packages with people outside the network.
I presume so. I'm not personally developing them. I know few people who are. I'm sure they're working closely with the DCP, the CVI, and so on.
But that's a good point. That can be very useful. That's great.
So if I can,
please.
I think the metadata is doable in terms of if the data are there. So the question is, does anyone have a diverse data set or on any of these axes?
Gender is okay.
I mean, we're starting to build on that with the support from CVI. Young pediatrics, but limited. I mean, it's 3 sites, right? So this covers a very small part of the world.
And in those 3 sites, what is the end?
The end, I mean, the target is, I think, something on the order of 30 each, right? This kind of thing.
And what percent do you think is European ancestry roughly?
The European industry would be 30%. Okay. Yeah,
Oh,
one
okay.
site European, then yeah, another site would be predominantly black, and another site predominantly this is in Pakistan. So yeah.
So that's a fantastic.
In the existing networks, but this we don't have this amount of diversity, at least from what we know.
We have like specific sites that are skewed one way or the other, but they're all in America. Right, so it's only there's no geographic diversity, there is genetic diversity.
Yes.
And considering the genetic diversity that you were mentioning, So, for example, especially in the example of the gut, you were mentioning that, well, it's important, for example, in London, where you have a huge immigrant community, so you can sample with a lot of different genetic background, right? But in terms of diet, and in the case of the gut, are you also including that in the metadata? Are you also including whether the person is a vegetarian or a vegan or a gluten-free? And is this affecting gene expression regardless of the genetic background? And how can this be included in the metadata? Maybe it's something that we should also
That's just increased it one level. No, but that's excellent. It's an excellent point. Yes, please.
Okay. So if you go to the human cell atlas data portal right now, I just looked up colon. Okay, we have Homo sapiens, like humans. We have age. We have gender, biological sex. We call it. We have donor level disease. So if they had like brain cancer, but their colon is healthy, and then you can look at methods of like processing and assaying.
So that's at the data level.
At the
level of the data set, yeah?
No, at the level of the donor.
At the level of the broken down.
Specimen, yeah, so just talking about the search filter, so going back to that, like our data resource will have minimal information about donor, like things like geographic location, that's going to be behind some sort of like metadata security where you have to access, like request access, and then if you have it, we can make it available. But there is some genetic, like some diversity available that's still safe enough to identify. Actually, no, we do have nutritional state. It really just depends if the data contributor gives it to us or if it wasn't, if we reach out and they never collected. Yeah, but we do have those fields and you will find them in some of the data sets. So we like smoking history, medical history, nutritional state, like it's extensive.
So then can we start to ask that from the tissue sources to get this kind of information? Because I agree, Ines, on your point of diet. Even smoking can affect the gut. So for gut sampling, it's not reported very often. For lung sampling, it always is because it's very obviously a factor.
Yeah. And with the geographic diversity also comes the microbiome, and how it affects gut and oral cavity, for instance.
Yeah.
We came up with a template for all metadata collection, which encompasses medications as well.
So this was really happy to engage in this. It's fun, yeah. But we need to wrap up soon. So just to finishing the core here, computation. Right. So we heard about it a little bit in the beginning, in the introductory talks about okay, we need X and we need Y, we need Z. But if you can really put the one single most pressing need that you can think of from that domain in your work, what would that be? There has to be something. It can't be perfect.
One thing, I guess, from my experience with the gut is in terms of QC metrics may be different depending on the cell type. So for example, we find epithelial cells and plasma cells sometimes get excluded even though they're good quality cells. And so it would be interesting to know how to approach QC in this way.
Yeah.
One place you can look is how other states are treating neutrophils. Because neutrophils tend to have 400 to 800 genes max. So they're typically going to look like garbage compared to the rest of the cell. So if you look into the literature around neutrophils, you should see different ways to handle.
Not mention the mitochondria RNA to see in there. Yeah. Yes, that's also what you see. Other challenges people think about the computation part.
The disconnect between the people who are doing the analysis and the scientists who are asking the question.
Oh, that's a big. What can we disconnect?
So, you know, like there's the computational people like for example, we do disease versus healthy, right? And then we do young versus old, right? Pediatric versus adult. And they give us one or the other, but they don't give us both. So we can't tell if the differences that we're seeing, old versus young, are because of health and disease. Like really basic, like just miscommunication of what is important and what are the questions that we're asking. We get what they think we will find important.
I think it's true that we lack tools with multivariate analysis.
Exactly, multivariate analysis, that's the, and somebody already mentioned the interaction term between geographic location and diet, and that just made it easy just to think. Okay, so,
Okay, so the plan is that at 6 o'clock, we're going to reconvene in the main hall. We're going to have an even faster report back than the last one. You know, my colleagues are telling me I talk way too fast. We'll try not to talk that fast. And then about 6:40, we're going to leave for the restaurant. What we're trying to do is get the meal over with before the thunderstorms hit around 9:30. So that's our ultimate goal today. So if you could just take a really quick break, and then 6 o'clock, another 30 or 40 minutes. And we'll
just first of